We have you here for 30 minutes, Minister. We'll let you speak to that as you wish, and if you're able to provide us some time for questions, that would be helpful as well. Thank you.
Sure. Kia ora mai tātou. First of all, thank you very much for the invitation to join you here this morning. Can I acknowledge some of the officials who are beside me here at the table and behind me. Professor Emily Parker, Professor of Chemical Biology at Victoria University and Departmental Science Advisor, and Simon Ray, Director of Policy at MB. So submissions closed on this bill last week, a bill that's looking to modernise the current gene technology settings, which is currently under 30-year-old legislation. And this bill fundamentally proposes a risk proportionate response across 4 domains: exempt, contained, environmental, and medical. I think there's reasonably good understanding around exempt, still some work to do. I think medical as well. I think in contained, but I think there's a lot of work to do in the environmental domain. That would be my expectation for what these submissions may show as well. The bill's modelled on the Australian Gene Technology Act 2000 and also aligns with our coalition commitments. And indeed, some progress was made under the previous government, including the report Whakaaronui, which was also leading to make some progress in this area. Finally, just a reminder, and then I'll conclude and actually move to questions, that this bill sets out a framework, that the detail will actually be in the secondary legislation. And I think we need to keep that in mind as well. But other than that, Mr. Chair, happy to take questions.
All right. Okay. Dr. Verrill.
Yeah. Thank you, Dr. Reti, and we're lucky to have someone of your expertise seeing this bill through. I want to start with a really high-level question about the logic of the bill before we dive into what is an extensive area of detail. And it's this. What are we regulating? Are we regulating processes or outcomes and risks? And I see elements of the bill that are trying to do either. And if I, to take a very, the working understanding you get of genetics as a medical professional, I recall learning that one small change, one nucleotide change, can lead to a total change in a protein's function. Yet parts of the bill seem to be saying, well, we'll view certain processes as low risk because they're similar to what could occur naturally. And of course, these one nucleotide changes do occur naturally. And yet the result of them can be quite significant. So that's what I'm trying to understand there. What are we regulating, the process or the outcome?
Yeah, I think it's a good question. I think parts of the bill do both. For example, we talk about gene editing, which is clearly a process and a technique. But if we look at the exempt category, it's actually to make decisions around the outcome. We say that those techniques that generate an outcome that conventional methods also generate, that will exempt them. So that's very outcome focused. But parts of the bill, as you described, do talk about process. Emily, would you like to talk to that?
And of course, there's some existing processes under HASNO Act that are exempt, like UV irradiation of organisms, which causes many genetic changes, and chemical mutagenesis is also exempt currently under the HASNO Act. So the intention is to carry that forward into the new Act as well. The gene editing provisions are outcome-based, not process-based.
May I just, if I could respond, one, of course, CRISPR has been developed since all of those acts were in place. So I don't find arguments about what the existing law is particularly compelling when we're dealing with a new technology. So continue to try work through this concern as we go on.
So what was the point you were making, that CRISPR has come on since HASNO? Was it? Yeah, right, 2012, I think, wasn't it? And HASNO was 94, I think. So I think this is part of what we're trying to update, trying to bring these modern technologies into account.
All right, now I'm going to go to Deborah and then Hamish, Steve, Jenny.
Just reiterate what my colleague Dr. Verrill said about having you in the hot seat for this one.
You're very kind.
Some of us, it's a really difficult field for a lot of us because we just don't have the understanding. So it's quite reassuring to have at least some people in this room who have at least some understanding of some of the difficulties there. Look, one of the things we've been told throughout is that the New Zealand legislation is, we're not trying to mirror the Australian legislation, but it's very largely drawing from that regulatory regime, which by and large seems to have worked reasonably well. So one of the things I'm unclear on is where our legislation or our proposed legislation differs from what they're doing in Australia and why we put those differences in. I'm assuming it makes it better, but if you could just speak to that.
Sure. Look, I'll talk to a few points and then again I'll pass to Emily, and you raised one of them in your first reading around SDNs, for example. So Australia does regulate SDN2. The intention of this bill is not to. And of course, if we look across the spectrum, if we look across the world in this particular area of SDNs, we can see the UK, for example, with their Precision Breeding Act 2023, which was enabled last year, it actually enables cisgenic SDN3s. So it's right at that extreme, if you like, and it's for animals as well as plants. The EU also looking in short notice to move to SDN3. We think that's a bit far, albeit cisgenic. It's a bit far for us to go. So we'll sit around the SDN2s and SDN1s for small gene editing sequences. So that makes us different in that particular area to Australia. We also have some differences around the management of ethics, for example. There is an ethics provision in the Australian legislation being passed to a ministerial council. My understanding is that hasn't actually ever been done, but we don't include ethics inside. We don't embed that in the bill. Emily, other changes?
So one of the differences, and Australia is just going through a consultation period about changes to their act at the moment, but and some of our advice was from their early consultation on that. So what is different about this proposed bill is to separate out and contained an environment and a medical one. People, there's been quite a big focus of people thinking that there's a big distinction between it. Actually, in my view anyway, it's a transparency thing about where activities are and how they're undertaken, rather than hiding them within a single layer, a single stream. So it actually makes no more difference to the regulation than would be having them in a single stream, but it clarifies them and is a transparency over what can happen under what conditions, as allowed by the different risk tiers of the bill. So we think it spells out, it's a complexity, but we think it is a good nuance for transparency in our environment. Probably the other change, Minister's the MAC as well. That has been included in this bill, and Australia, of course, don't have.
Sorry,
the
the what? Did you say that?
The
Maori Advisory Committee.
Oh, right. Thank you. Yeah. May I have a sup on?
Sure.
So, thank you. That's helpful explaining where the differences are. I do want to go back to the ethics one. And it does concern me. When we spoke with the officials, they assured us that a whole lot of the ethical considerations were considered within existing ledge among animal welfare. And obviously anything we're doing with human beings gets considered by ethics committees and the like. What struck me about that was very much focused on a welfare analysis and the welfare of organisms, of human beings and animals. It concerned me that there wasn't particular attention being paid to other sorts of ethics. So in particular environmental ethics, and the sorts of ethics we might have concerns with around ecosystems. And so the ethics of operating with various types of plants. Now, we don't typically worry about welfare concerns with plants, but there are a whole set of other ethical concerns and around ecosystems and the like. And so it concerns me that the focus of ethics was so narrow within this bill that it didn't consider those other sorts of ethical concerns which are well established within environmental ethics and the like. This might just be one tiny space where my own expertise might come to play here, that I do. There's some Kantian types of ethics or that we might want to consider in this space, and yet there is no ethics input in the system anywhere that would deal with some of those sorts of issues.
So two things, correct me if I'm wrong, Emily, but I don't think currently has an ethics component to it. So what we do at the moment doesn't embed in legislation an ethical. That doesn't mean that it shouldn't. I'm just stating the status quo. And your point is well made. I don't actually know what the answer is around environmental ethics. And do you have any?
So I mean, someone might be able to answer this, but I draw attention to the definition of environment. in this and what needs to be considered, and if you believe that if you think that's fit for purpose in managing some of the issues that you're thinking you're raising.
So let me just reframe that, Simon. Is there I mean, we know where to point human ethics towards when we have questions. Where would we point environmental questions?
Yeah, I think Emily's right. I think it's about how we think about the environment and environmental risk, and the way in which the environment's defined in the Act, and the way in which harms to the environment or risks to the environment are considered in the Act, and how that would flow through in the risk management framework that was adopted by the regulator.
Can I have a sup on that possibly, Mr. Chair?
Yes, yeah, of course, and then I'll go to Hamish.
Yeah, thank you. Given the broad nature of the possibilities with genetic engineering and gene editing, it spans across food production, environmental use for perhaps pest management, medical use and application, in containment, precision fermentation for food, animals, plants, insects, you name it. Surely there are a multitude of forms of genetic engineering coming down the line that we cannot conceive of. And it is exactly for that reason that perhaps the Australian version of legislation did have an ethics consideration, because we can't imagine all the permutations and the impacts, not just on the environment, but on perhaps agricultural producers for certain application, or on food types, or on wild animals, on animals in containment. Are you open, Minister, to seriously reconsidering whether there should be an ethics component to the evaluation of new organisms?
Look, I'd have to say it's not part of my current thinking, and correct me if I'm wrong, have the Australians, and Simon, you may notice, have they ever utilised that? I think it's referral to a ministerial council, I believe, is what the Australians do.
So the Australian ethics provisions are not operative in the decision making. So they have a separate ethics council that advises the regulator on general issues to do with ethics. The regulator doesn't take ethics into account in its decision making.
Right. So when does that kick in?
So essentially what we understand is that this might be something we could go into in more detail in closed hearing, I think.
I think what I'd say to you, Steve, is it's not currently part of my active thinking, but submissions are so that we can take on a wide range of views and ponder and make the best decisions. So not currently part of my thinking, but let's see what submissions and others say.
Quick question.
We're going to Dr. Campbell.
Excellent. Thank you, Minister, for coming and speaking to the Select Committee. Can we just take a bit of a step back? What's the problem we're trying to fix, and what issues do we have with the Has No Act?
Sure. I mean, the problems we're trying to fix, as has been mentioned by the team here, is that the legislation is 30 years old. It doesn't take into account new technologies. For example, we've mentioned CRISPR-Cas, if you want to call it that, as one of them. Secondly, it's not specific to gene technology. It's hazards as well, and we think there needs to be a specific focus in legislation around hazards. We think a risk proportionate approach is useful as a way forward, where it's a bit more blanket under Has No at the moment. So the problems we're trying to fix fundamentally are to modernise and bring us up to date with where other jurisdictions are with gene technology is what we're trying to do with this bill fundamentally. Yeah.
Okay.
What other jurisdictions?
I had Steve and then I
Who's
had
next?
Jenny.
Next.
No, I mean,
He's already had questions.
I just had a sup. We just sup.
Well, mine's a sub on that.
I'll be quick. Minister, there's been a couple of submissions from industry who have pointed out just how exposed we are as an exporting nation to trade and market concerns. The Dairy Companies Association says New Zealand's majority primary sectors, dairy, meat, fisheries, wine, forestry, selected horticultural products, export up to 95% of their output, not a level matched in other developed countries. So their concern is that we are highly exposed to market and trade considerations. Our primary industry is uniquely dependent on overseas trade. The Ministry of Foreign Affairs and Trade recommended that there should be a trade and market access consideration of legislation. Is that not a very sensible thing to do for a country that is so dependent on overseas exports, where there is clear concern in overseas consumers around genetic engineering?
So this is a good question. I think if we look at the Australian Gene Technology Act, it doesn't take into account trade considerations. So if we are modelling on that, then they haven't seen it worthy to do that, first point. Secondly...
But we're not Australia, are we?
No, but we are modelling the bill on their act. It does form the core of how this bill comes to be. But I take your point, we're not Australia, and there are some differences that we've already talked about. Secondly, I think, and the point you're making is we are so dependent on primary sector exports that we're different to others, because otherwise it would be easy to say, well, look at other vibrant economies that have brought in gene technology, and the sun is still risen in the east. There's still been an organic and a GE-free, a vibrant sector. The point you're making is we're different because we're so primary sector dependent. That's the point you're making, eh? Yeah. Yeah, look, I have heard that discussion. I'm still working that through. It doesn't currently sit inside the bill. Again, I'm amenable to submissions and discussions on that, but it doesn't currently sit in an active work stream with me. Yeah.
Judy.
Modernising what we do in medicine, can you give us a layman's speak about what this bill will do in terms of medicines and off into the future? Some sort of
Sure.
future
I think, I mean, the quintessential example would be Malahide Institute and their CAR T cell therapy, and I believe you may be meeting with them later today. I mean, that's a great example, I believe, of the 3 GMOs released into the environment under HASNO. I think Malahide's had 2 of those 3. Their initial 2022 application that gave them permission to modify the T cells, and then their 2024 application that let them go to clinical trials. You know, this is an exciting piece of research, an exciting endeavour, and at the moment we're cutting edge in this technology. Seems like we can point these killer T cells at precisionally defined cancers, and amongst other things, get less side effects than other remedies and other solutions. This is an exciting thing. They're into phase 2 now, looking to enrol 80 people. I think they enrolled 8 as of 2 weeks ago. This is leading edge. This is sort of the definition of precision medicine, where you can specifically identify a treatment just for you, just for your cancer, just for your B-cell lymphoma. These killer T cells can attack that. Now, without the advances that we're looking to make in this bill, and Malahide may go over this as well, but some of their approvals took a year to get through. I believe the first approval, maybe it was the second, took a year to get through at a cost they estimate of about $250,000. That's not insignificant. They were able to progress, but again, maybe they'll raise other examples of international studies that they wanted to be a part of, and because of our restrictive regime, they either had to pull out or not able to participate. And they've raised the question with me of reputational damage to our science sector by not being able to participate in those sort of studies. So real opportunities in the medical and health space for what a modernised gene technology bill could do. And I just give one example with CAR T cells.
Thank you. Minister, the submission from we have received advice that perhaps the provisions in the bill could be more clearly articulated to reflect policy intent around non-regulated technologies. Are you open to having feedback from the committee on that area?
Yes.
Thank you. Well, we've heard about some of the advances in healthcare. What are some of the other examples, maybe more broadly, and what are the economic benefits that New Zealand could reap?
So a wide range of examples. Emily, you might want to talk about the containment space, which I think is your particular area of expertise, because often it's one not, we talk about medical, that's got some visibility, environmental. Clearly we could go to some of the ag research stuff and the scion stuff. The containment's less visible. Do you want to talk about that?
So some of the research, precision biomanufacturing, for instance, where yeasts or other organisms are engineered to produce proteins of value. So the output of that that process. Is a valuable protein, for instance. Some of we currently are under significant volume restrictions around that, even when it takes place in containment, and that really curtails activity. We've examples, perhaps of Daisy Lab startup in Auckland in this area, who that has been a huge barrier to success of as well. So there's a whole range of technologies that have come on board, which there are barriers which are disproportionate to risk that this bill is trying to address.
Excellent. Thank you.
Cameron.
Thank you, Mr. Chair. I just had a couple of questions. We asked the officials previously around what the definition of indigenous species around native or endemic or indigenous. And I was sort of thinking about it for a while and received some advice back. It hasn't really cleared it up as much for me. I would wonder what would be the potential of having an exhaustive list of species that are considered applicable to the section of the Act, section 128, where a kaitiaki relationship can be asserted. So is that a possibility in this? What do we think about having an exhaustive list?
I'll pass that to Simon, but I asked the same sort of questions and was indicated that indigenous species means a species or organism that is endemic or native to New Zealand, and that wording comes from the Plant Varieties Act 2022. So I also was exploring what does this kaitiaki relationship look like? What does it mean? The question you're raising is whether there should be identified species that sit inside that kaitiaki relationship. Simon, do you want to talk to that?
Yeah, so we haven't attempted to do that, largely because our advice from our Māori focus group has been that it's very difficult to set that out in advance. Sometimes it's contested, and generally speaking, the process of establishing a kaitiaki relationship is done through tikanga. And so what the bill does is it opens up a space whereby you can have a conversation about tikanga and establish that the kaitiaki relationship exists. And then that gets considered by the regulator.
That's fine, but the bill does specifically say indigenous species. So, I mean, there's a tikanga aspect about figuring out where the kaitiaki relationship is, but that doesn't change the species that exist on the ground today. And that I wouldn't have thought. Where is the classification? Are we going to allow are we going to have the ministry just leaving it open to what is an indigenous species? Are we going to create, I mean, if you're saying that we need to decide what's indigenous, native or endemic based on a kaitiaki post process, is that what I'm hearing?
Yeah, I might pass this to Emily because Emily does work with indigenous species, so she will be able to give you a bit of an idea of the difficulties of describing listed.
Yes, I mean, we're very aware that some of our organisms that even are endemic, the sequence variation is very close to sequences that are seen in other parts of the world and species in other parts of the world. So it's defining that kaitiaki relationship is important, that connection is important too. So it's quite hard to define specifically what is in that category versus what's not. So the definition of indigenous takes that into account to allow kaitiaki relationships to be established.
I must say your answer does blow it out even further than you just. Like you're basically saying we haven't got any definition. We can't define it. We will leave that up to discussions later on as to what. And so, I mean, when someone reads the word indigenous, you think, okay, that's fine. It's species that are, I don't know, potentially only found in New Zealand or were here before a certain date, perhaps.
Mm-hmm.
But what your answer is just elucidated as something that we don't even have that as a definition.
So I think the difficulty is there's a very long list of indigenous species. It's a very large number of species.
So the data capacity being built around the world to store that sort of information, why would that not be something we could do?
But to have that information, I mean, that would be across plants, microbes, animals. That information isn't even available to us.
All right.
So species haven't been classified since Aristotle.
Especially in the microbial space, unfortunately.
Cool. We've got 4 minutes left. Steve, did you have another?
Yeah, I've got a question around traceability, Minister.
Dairy Association, also Beef and Lamb and others have expressed real concerns about the responsibility for ensuring traceability rests with gene technology organisms through the supply chain entirely rests in the Animal Products Act or the Commercial Traceability Insurance Schemes. And that without a regulatory underpinning by the regulator at the start of the process, when the decision is made to release an organism, then the onus and the cost will fall to all of those non-GMO producers to try and account for their status if they are marketing a product as non-GMO. And there's real concern from industry about, frankly, the unfairness of that and also the difficulty of traceability if you have organisms that are exempt from any regulatory oversight.
Simon, do you want to talk to that?
Yeah, I think there's quite a lot of complexity in terms of how traceability works in practice. And there are some decisions to be made about the appropriate place for costs to lie in the system. And that tends to vary from product to product. And I think that's something that, the Minister's indicated he's interested in what the committee thinks and where the submissions go on that. And I think it's something that will be good for us to dig into in greater detail.
So you're open to tightening that up and considering proper traceability?
Certainly open to the discussion, yeah.
I've got another one if there's time.
2 minutes. Got 2 minutes and that's it. No.
Yeah, I mean, one other thing that Beef and Lamb pointed out, that they applied for an extension, the Meat Industry Association, Dairy New Zealand and Dairy Companies Association, they all felt it was quite a rush process, this. They were not able to consult with their farmers and their farmers weren't happy about that. What is the rush around this when a more substantive, thorough process that allows these very significant industries to properly engage with their members, why is it being so pushed through? And is there not a serious risk of public discontent and industry discontent on something so significant?
So several things. There is a willingness from government to progress this, first of all. Secondly, 15,000 were roughly able to complete the process on time. And thirdly, when I look back at the hazardous substances and new organisms, open bracket, genetically modified organisms, close bracket, amendment bill that came in 2001 was almost exactly the same consultation period, 14th of December 2001 to 1st of February 2002, for a very important bill that looked to add genetically modified organisms to HASNO. So very similar sort of piece of work, albeit the number of submissions were quite less. So at the end of the day, it's actually for the committee to decide when submissions open or close. But I am indicating that we want to progress. It's not actually a call that I make.
Okay, so you're not averse to us taking a bit more time with it?
It is a committee decision, but I am indicating that 15,000 were able to get in on time and we do want to progress.
All right. We're at time. Hey, thank you, Minister.
Thank you very much.
All the
Thank you for what you have
everyone.
done, what you will do as you go through these submissions.
Thank you for being here.